So we spend about 1/3 of our life on sleep, but the roles of sleep are, I would say, far from clear. So what I'm going to introduce today is our research on sleep and learning. So traditionally, sleep has been thought to occur uniformly in the brain, but it is now known that sleep can occur regionally. And one of the extreme examples of non-uniform sleep is known as unihemispheric sleep in marine mammals and migrating birds. So they can show wakefulness in one of the brain hemispheres, and the other brain hemisphere can be deeply asleep. And we found that even humans can show asymmetric sleep, and this asymmetric sleep can occur in a new environment when we suffer from sleep problems, known as the first night effect. I'm going to come back to this topic a little bit later during this presentation. So as you can see, we animals can show sleep in various forms. Then my question has been, why do we sleep at all? So today in this talk, I'd like to present some results related to how sleep might contribute to the changes in the brain plasticity and learning. Okay, so in this seminar, I'd like to introduce two topics. And I'll start from the first study where we asked how do NREM and REM sleep play a role in visual perceptual learning in humans. And we specifically focused on visual perceptual learning, or VPL, in the study. This is a visual perceptual learning is a long-lasting performance improvement on a visual feature after visual experiences. Visual perceptual learning is known to be prevalent and occurs continuously in our everyday life. And as our perceptual system adapts to this world, we become better at detection or discrimination of different visual stimuli. For example, by intensive training, determining chick's gender, or finding pneumonia on an X-ray picture, which these are usually very difficult, but become possible with training. And once you have this increased sensitivity in your brain, then you can keep this increased sensitivity for over many years. So visual perceptual learning has been thought to represent plasticity in the visual system in adults' brain. And with training, as I mentioned, you will have better sensitivity to various low-level visual features. For example, people can be trained to better discriminate the differences in the orientations of the stripes. These are the examples of the visual stimuli used in experiment. And this effect of learning does not easily transfer to untrained feature, including visual location. For example, if you're trained with an orientation in one of the visual fields, say upper left visual field, then the training effect does not easily transfer to untrained visual field, such as upper right visual field. And this is known as location specificity. And visual behavior and neuroimaging evidence has demonstrated that early visual areas are involved in visual perceptual learning. And this is in a reconstructed image of the brain. And this is front, and this is back of the brain. This is top and bottom. And we're looking at the brain from the medial part. And this circled in yellow, this part is early visual areas. And recently, it has been shown that learning consists of multiple different phases. And the first phase is acquisition. This is where people are actually engaged in a task in the training session. And it is known that learning is still fragile right after training, so it can be easily disrupted by training on another task. But learning does not end at this acquisition stage, but it is processed further offline. And the next phase is known as consolidation. This is where learning becomes more and more resilient to training on a new and different task. And this consolidation had been known as time dependent, meaning if there are several hours of time between training on the first task and the second task, the first task, learning of the first task will be more robust against training on the second task. And interestingly, accumulating evidence indicates that important processing takes place specifically during sleep. But how sleep is involved in learning or performance improvements remains unclear. Okay, so here I'd like to summarize some characteristics of sleep stages in human. And this is an oversimplified transition of human sleep, and which is called a hypnogram. And in this figure, x-axis is time and y-axis is sleep stage. And this is wakefulness, this is REM sleep, and these are non-REM sleep, and N1 is the lightest sleep, and N3 is the deepest sleep. This is also called slow wave sleep. And so human sleep consists of two different sleep stages, REM and non-REM sleep. And we actually repeat this non-REM and REM sleep cycle for about 5 to 6 times a night. And this shows an example EEG waves of non-REM sleep. The waves, these EEGs during non-REM sleep are synchronized, and the key brain oscillation, sleep spindles not to be involved in learning and memory occur during non-REM sleep. And this figure shows the an example EEG wave during REM sleep. As the name indicates, rapid eye movements occur during REM sleep, and EEG waves are desynchronized. And importantly, REM sleep usually follows non-REM sleep in normal sleep-wake cycle in human sleep. Now, there are two important aspects in the role of sleep in visual perceptual learning, and one of them is offline performance gains, and the other is learning stabilization. So earlier studies have shown that performance on visual perceptual learning is improved or changed dramatically beyond the baseline level after sleep. So since this significant improvement in performance occur between sessions without additional training, such changes in performance are called offline gains. And when people are deprived of sleep after training, then it is also known that these offline gains are gone completely. And this figure shows important finding by Stickgold et al. And the black bars show performance change over multiple days after training on day 0. And this white bar shows performance change when participants were deprived of sleep on the night of the training. So as you can see, even after 2 nights of recovery sleep, the effect of learning is diminished if participants were deprived of sleep after training. So this shows that there's an important time window for sleep to occur after learning. And interestingly, sleep is also implicated in another process involved in stabilization of learning. So as I mentioned briefly earlier, it is reported that the learning state immediately after training is fragile, so it can be disrupted by learning on another task, unless there's a certain amount of wakefulness before new learning takes place. And also, a previous research have shown that 1 hour of wakefulness or overlearning make learning resilient to interference. Additionally, it has been shown that if sleep occurs after acquisition of learning, then learning becomes resilient to new learning after sleep. So this suggests that sleep also plays a role in stabilization of learning. So, so there are 2 stages of sleep, non-REM and REM sleep. Then how are these different sleep states involved in learning? And previous studies have found that after training on the visual perceptual learning task, early visual areas increased its activity, and increases in activation was correlated with the amount of offline performance gains. That is, the performance changes over sleep. For example, fMRI signals increased in early visual areas during non-REM sleep and was correlated with offline performance gains. And also, brain oscillations, including sleep spindles, are increased in the trained visual location, visual field, and then the amount of increase was also correlated with offline performance gains. So non-REM sleep may be involved in making learning more plastic. And however, REM sleep has been also found to be involved in visual perception learning. The study by Carney investigated whether deprivation of a particular sleep stage has an impact on visual perception learning. So in this figure, the y-axis shows performance improvement after sleep. So larger values indicate better performance. And the x-axis represents each subject. After normal sleep without sleep deprivation, subject showed significant performance improvement. Also, after slow wave sleep was deprived, this is a type of non-REM sleep, deepest non-REM sleep, and subjects still showed improvement after sleep. However, after REM sleep deprivation, the significant performance change which occurred in other two conditions was gone. So this result indicates that REM sleep also plays a certain role in visual perception learning. However, how REM sleep is involved in learning remained unclear. So how do non-REM and REM sleep play a role in these two different aspects of learning, that is performance gains and stabilization learning? As I have mentioned earlier, non-REM sleep plays a role in offline performance gains. So non-REM sleep may make learning more plastic to enable behavioral changes over sleep. And importantly, this process alone does not explain why our learning can be maintained, because if there's only one process that supports offline gains, a learning state can be kept highly plastic. So learning could be disrupted by training on another task upon waking. Then we came up with a very simple hypothesis. Since REM sleep usually follows non-REM sleep, then REM sleep may play a role in stabilizing a learning state which becomes highly plastic during non-REM sleep. And this is a very simple hypothesis, but we decided to test this. Okay, so we first ask this question: is REM sleep involved in stabilization while non-REM sleep is involved in offline performance gains? And subjects were trained on a texture discrimination task, or TDT. This is a very standard task in visual perception learning. And the figure shows the main stimulus display in TDT. Subject had two tasks. In the main task, subjects responded with the orientation of the target triplet, which was presented at the periphery of the display, was aligned vertically or horizontally. In this case, it is vertical. And the other task was a letter task. Subjects responded whether the center letter was L or T, presented in the center of the display. And in this case, it is a letter L. This letter task was to ensure subject fixation at the center of the display. And this shows one trial of TDT. In addition to the stimulus display, there is a mask display to vary the task difficulty. And these two displays were presented in an interval known as stimulus to mask onset asynchrony, or SOA. And when the SOA is shorter, the task becomes more difficult. So we presented many trials with various SOAs, and we measured subject response accuracy for each SOA, like this. And then we plotted a cumulative Gaussian function to obtain the threshold SOA, at which 80% accuracy is marked. For example, in this case, it is, let's say 170 milliseconds. Then the threshold SOA gets shorter, usually by training. For example, we might be able to plot the data like this, and then it may the threshold SOA may become 130 milliseconds. And then what we did was to measure this threshold SOA at each of the test sessions, and then we obtained the change in the threshold SOAs. For example, in this case, there's a 23% improvement in performance. Okay, so using TDT, we implemented an interference paradigm to investigate the stabilization learning. And the key element here is the orientation of the background lines of this stimuli. So learning of TDT is specific not only to the trained retinal location shown here, but also to the orientation of the background lines shown here in this picture. And in this case, the orientation of the background lines is horizontal. So these are aligned in the horizontal lines. Now, if the orientation of the background lines is the same across the two training sessions, say horizontal, then horizontal again, learning occurs. However, if the orientation of the background lines is changed from the first to the second training without time interval, say from horizontal to vertical, then no learning occurs with the first background due to the interference from the subsequent new learning on the first learning. This interference occurs because learning on the first task is still fragile. And it takes some time to be stabilized before training on the second task. So using this interference paradigm on TDT, we investigated whether REM sleep stabilizes learning. So if REM sleep occurs between these two training sessions using different TDTs, and if REM sleep plays a role in stabilization learning, then second task no longer should disrupt the first learning. Therefore, learning of the first task should remain even after learning on another new task. However, if REM sleep does not occur between trainings, and if there's only no REM sleep between these two, then interference from the second task on the first task should occur. So learning on the first task should be disrupted due to this interference. Okay, so all the participants were young and healthy adults. They had a regular sleep wake habit. We made sure using actigraphy watch and sleep log, and also a bunch of sleep-related questionnaires. And we also had an adaptation session before the main experimental sleep session to control the first night effects. In the same series, Dr. Andrew Lon has kindly introduced our study. So in his Being Awake While Sleeping seminar. So I was just going to briefly mention it here. When the first night effect occurs when we sleep in a new environment for the first time. And for example, when we are on a business trip and when we had to sleep in a new hotel room, then it may take a while to fall asleep, and we sometimes wake up in the middle of the night for no reason. And these are the examples of the first night effect. This first night effect occurs also can mess up our sleep studies, because when we invite participants to sleep in sleep chambers, and we want to get good quality data, but actually people are not really used to sleep in sleep chambers, so the sleep quality declines, and we sometimes have to throw away this data. because of poor quality of sleep. But we also found in the studies that when people suffer from the first-hand effects, this depth of sleep showed asymmetry between the left and the right brain hemispheres. And the lighter sleeping left hemisphere was actually monitoring the environment. And when the left hemisphere catches rare auditory stimuli, then people can actually wake up and respond faster to the sounds. And so we actually had a chance to work with TED Ed, and they kindly made a video which is on YouTube. Can you be awake and asleep at the same time? And it's really interesting. So please take a look when you had a moment. So why we wanted to control this first effect is that we don't want to see asymmetric sleep like in marine mammals and birds. So now this shows the experimental design in the present study. And after training on the first task, there was an interval when subject had a 90 minutes nap. I will explain this MRS later during this presentation. And after this interval, training on the second task was conducted. And we measured performance change by sleep by test sessions conducted before and after nap to measure the amount of performance gains. And we measured the amount of stabilization learning by the test sessions conducted before and after the second task. And subject were further divided into NREM and REM group and the NREM only group, who showed no REM sleep without REM sleep to investigate the effects of REM sleep on learning. Now I will show you the performance results. This figure shows the result of offline performance gains, and larger values indicate better performance. And in both NREM and REM group and NREM only group, performance improved significantly after sleep, and there is no significant difference between these two groups. And this figure shows the performance change after the second task. And so this shows the amount of stabilization learning. Larger values indicate more stabilization, and smaller values indicate less stabilization of learning. And for the non-REM and REM group, performance significantly improved after sleep. However, for the non-REM only group, even after the training and the sleep, performance deteriorated significantly after training on the second task. Now, because the number of data was still quite limited, we conducted another experiment with larger number of samples. In here, performance on the first task was tested before and after training and sleep. So again, larger values indicate more stabilization, and smaller values indicate less stabilization of learning. And when for the non-REM and REM group, performance improved significantly after training and sleep. So there was no effect of interference from the second task. But for the non-REM only group, even after training and sleep, there was no improvement after the training on the second task. So this shows that stabilization of learning did not occur without REM sleep. Now, some of you may wonder what happens to the training on what happens to the performance on the second task. So this shows the result of the second task, and by comparing the test sessions conducted before and after sleep. So as you can see here, if there was both non-REM and REM sleep during this nap session, then learning on the second task, which occurred after sleep, improved significantly. However, when there was only no REM sleep, without REM sleep, there was no significant improvement even after training on the new task. So this shows that REM sleep not only stabilized old learning that occurred before sleep, but also enabled post-sleep new learning. Okay, so our next question was, does non-REM sleep increase plasticity and REM sleep decreases it in early visual areas? So what we wanted to measure was the degree of plasticity of the human brain in vivo and magnetic resonance spectroscopy, or MRS, was useful. So we decided to use this. And MRS is a non-invasive imaging technique to quantify neurotransmitters, the amount of transmitters, including excitatory neurotransmitter glutamate and inhibitory neurotransmitter GABA. So the volume of interest was early visual areas. And earlier, we established that the concentrations of glutamate to GABA ratio, or the EI ratio, was strongly related to the degree of plasticity of early visual areas. For example, the EI ratio was increased after training and decreased several hours later. And also, these increases of EI ratio correlated with the fragility of learning, and decreases correlated with the amount of stabilization of learning. So, so using this MRS and measurement of the EI ratio, what we wanted to do was to measure these during sleep. So we decided to measure MRS and polysomnography at the same time simultaneously during the nap session. And we measured the glutamate and GABA during non-REM and REM sleep, and we averaged by sleep stages and normalized by wakefulness. Okay, so this shows the result of the EI ratio, and the y-axis shows the EI ratio change from wakefulness. Larger values indicate more excitatory, and smaller negative values indicate more inhibitory during sleep. Okay, so the EI ratio was significantly larger than wakefulness during non-REM sleep, and we found that the EI ratio during REM sleep was significantly more inhibitory compared to wakefulness. And next, we asked whether these changes in the EI ratio during sleep were learning specific or not. So we conducted a no learning condition without training before sleep. Okay, so this shows the experimental design, and this is the one I already showed you. And here, for the no learning condition, there was no training before or after sleep, and we simply measured the MRS and the sleep status during nap, and we compared the data between these two conditions. Okay, so the figure on your left is the result for the learning condition. So this is exactly the same as before. And the right figure is the non-learning condition. And for the non-REM, the EI ratio changed during non-REM sleep. Even without learning training before sleep, there was a significant change in the EI ratio. However, for the EI ratio changed during REM sleep, there was no significant decrease without training before sleep. So this EI ratio decrease during REM sleep was learning specific. Now, so the EI ratios are measured by glutamate and GABA concentrations. And we also asked what contributed to this EI ratio changes during sleep. So this shows the concentration changes for the glutamate and GABA during sleep for the learning and no learning conditions. And for non-REM sleep, there was a significant decrease in the amount of GABA during after learning, and also when there was no learning. See. And there was no significant change for the glutamate concentration. So the EI ratio change during non-REM sleep were due to the decreasing GABA level. This shows the glutamate and GABA concentration changes during REM sleep. And for the GABA concentration, there is no significant change with or without learning. However, for the glutamate value, there is a significant decrease in the amount of glutamate only after learning, and there was no change without learning. So the EI ratio decrease, which was specific to learning during REM sleep, was due to the decrease in the glutamate concentrations. Now we finally ask, are plasticity changes during NREM and REM sleep associated with learning? The left figure shows the scatter plot between the EI ratio change during non-REM sleep and the amount of offline gains by sleep. And the gray dots are on non-REM only group, and the red dots are non-REM and REM group. There was a significant positive correlation between them, meaning when the EI ratio is larger during non-REM sleep, then the performance, the amount of offline performance gains are also larger. And this shows the scatter plot between the EI ratio change during REM sleep against the amount of stabilization learning. There was a significant negative correlation between them. So when the EI ratio was more inhibitory during the REM sleep, then this would make learning more stabilized after sleep. So these results suggest two distinctive mechanisms for non-REM and REM sleep with opponent patterns of neurochemical processing. In this figure, the y-axis represents the plasticity level of the EI ratio during sleep, and the x-axis is time, and the left part is non-REM sleep, and the right part is REM sleep. So the this red line represents the EI ratio change during sleep after learning. We think that one mechanism is plasticity increases in EI in the early visual areas during non-REM sleep, and the other mechanism is the stabilization that occurs during REM sleep. So during REM sleep, learning is stabilized by decreasing once increased plasticity during non-REM sleep, as shown by decreases in the EI ratio below baseline. Furthermore, we found the process that was specific to learning. This blue line shows the EI ratio during sleep when there is no learning before sleep. During non-REM sleep, the EI ratio increases in no learning condition also, and was not significantly different between the two conditions. However, during REM sleep, the EI ratio decreases occurred in a learning dependent manner. So altogether, previous studies have suggested only one of non-REM REM sleep is more involved in learning, but what we believe is that non-REM and REM sleep both play important roles, but in different but complementary roles, at least in the case of visual perceptual learning. Okay, so now I'd like to move on to the second study, where we asked, do reward and sleep interact for visual perceptual learning? So far, I have introduced that there's the effect of sleep on learning, and actually there's another line of study that found that pairing of a visual feature and rewards facilitates visual perceptual learning. In this previous research by Seitz et al., subjects were passively viewing orientations of stimuli presented in the center of the display. There was no active task that subject had to engage in during the training session. It was just passively viewing. And there were two conditions, there were two orientations. And only one of the two orientations was paired with water reward, and there was no water reward given to another orientation. Subjects were deprived of eating or drinking 5 hours prior to the training, so this procedure made water reward effective. And this, the figure on your right shows the percent correct for the sensitivity tests conducted before and after training. And so as you can see here, pairing of a visual feature and reward actually improved learning. And there is no performance change for the orientation that was not paired with reward. And importantly, the training was always conducted across over multiple days for about a week or so. So subject had a night of sleep every time after rewarded training. Given that sleep is beneficial to learning, we wondered whether reward processing and sleep interact with each other to facilitate learning. So here was the question we asked in the study. Do sleep and reward interact with each other on learning? And if these are independent, then performance improvements by sleep and reward should occur in parallel. On the other hand, if they interact with each other, performance improvements with reward may occur only after sleep. Okay, so subjects were trained on a texture discrimination task as in our previous research. And for the reward groups, reward was a drop of water as in the previous study. And we used a tube inserted to the subject's mouth, and subjects in the reward groups were deprived of sleep or water for 5 hours prior to the training sessions to ensure that this water was effective enough. And during training, they received a drop of water as a reward upon correct response on this texture discrimination task. This shows the experimental design. There were 4 groups in total, and a sleep and reward group or a sleep and no reward groups performed this TDT in the evening, and they had an overnight sleep, and they came back to the lab to do retest session in the morning, next morning. And for the wake groups, they were training in the morning, and they came back to the lab after 12 hours of wakefulness, and then retested in the evening. And actually, there was one participant in the wake group accidentally took a nap, so the data was removed. Now, here are the results. The figure on your left shows the average performance improvement. After sleep, larger values indicate better performance. And this is sleep with reward and sleep without reward. And this is wake with reward and wake without reward. And there was a significant two-way interactions. For the sleep group, there was a significant difference with and without reward. And there was also a difference between the sleep and the wake groups with reward. And so there was no significant difference between the wake groups with or without reward. And some of you may wonder whether reward given before an interval, either sleep with either sleep or wakefulness, had already had an impact on learning. But that was not the case. And the figure on your right shows the performance during initial training. And there was no significant difference between these conditions. So our result demonstrated that there is a significant interaction between the effects of sleep and reward on learning, and reward is effective only after sleep. Now we next asked, what is an underlying mechanism for this interaction of reward and sleep on learning? So we decided to do sleep experiment, and there were two conditions. In both groups, there was a 2-hour nap period, but with the nap and reward group, there was no reward during the training session, and for this group, there was reward during training, and there was no reward for the no reward group. And the performance change was measured by test sessions conducted before and after sleep for both groups. Okay, so here this shows the behavioral test results, and so in both groups, performance improved after sleep, but there is also even larger improvement when people were trained with reward. So then how about brain activities during sleep? And the frontal central EEG electrodes, we selected these electrodes because previous studies indicate that there's a reward system that forms a neural arc that originates from the striatum to medial prefrontal areas. And also, there are previous research that the reward processing in the medial prefrontal area will appear in the frontal electrodes. So we decided to choose some of the front central electrodes as activation of the reward processing. And for the visual processing, we selected the in the trained and untrained side of the occipital electrodes. The trained side was the contralateral side to the trained to the target presentation of TDT, and the untrained side was the ipsilateral side to the target presentation of TDT. Then we conducted a frequency analysis to obtain the power of brain oscillations specific to the sleep and learning. Okay, so here, this shows the result of power density measured during non-REM sleep in delta, theta, alpha, and sigma activities. And each bar corresponds to the region and reward condition. It's a bit complicated, but this is prefrontal with reward and prefrontal activity without reward. Untrained visual field, untrained visual region, and untrained visual region. Untrained visual region with and without reward, and this is a trained visual region with and without reward. So one of the main results from this is that we found the sigma band activity, which reflects the strength of sleep spindles, found to be involved in memory processing during sleep, was decreased in the untrained visual area by reward during non-REM sleep. Now, this shows the result for the power density measured during REM sleep. And one of the main findings for REM sleep was that increased activation was found in the prefrontal regions with the reward, and it occurred globally across the frequency bands. So we next investigated whether these activities are related to learning. And for the trained and untrained power densities in the visual processing during non-REM sleep, this was correlated with amount of line performance gains. And for the frontal part densities, which would reflect the reward processing, this shows the also correlation with offline performance gains for REM sleep. Okay, so to conclude, rewarded training enhances visual perceptual learning through both non-REM and REM sleep, where the reward and the visual processing interact with each other by potentially removing unnecessary activity in the untrained side and strengthening the reward processing from the prefrontal area during sleep. So, okay, with that, this whole study was conducted with the at Brown University with many wonderful researchers, Yuka, Takeo, Ed, Gian, Tyler, Aaron, Deanne, and Jesse, and it was great working with all of these. And okay, so with that, I'd like to take any questions. Thank you very much.